Thank you very much, Nick. When we think about vaccines, what we really need to think about very carefully is immune memory, because in most cases, that's really what we're trying to induce. So during vaccination, we will get a textbook immune response with a small IgM response and even smaller IgG response. And what we hope to get at the end of this process is an immune memory, which then upon infection can be re-stimulated to get the classical secondary response, which is fast, has a high magnitude, mainly IgG, is persistent over a long period of time, and with an increased avidity. So really what we are after in most cases for a vaccine to be successful is to induce a long-lasting immune memory. And many people actually look at this response here and forget to look at immune memory. And what we have done is to try to investigate the quality of this immune memory response. And we have looked at the quality of the response in terms of a challenge with a number of pathogens which could be immuno-modulatory. And we have examples of viruses which can make homologs of cytokines, for example, or chemokine-binding proteins, or bacteria that has the ability to modulate and induce cytokines in macrophages or induce T regulatory cells, or parasites such as schistosomes, which can affect the Th1, Th2 balance, or leishmania, for example. And there are many examples of those so-called immunomodulatory pathogens. And what we like to do is to look at adjuvants in their ability to induce immune responses that will be able to withstand regulation by challenging pathogens. And so what we try to do is, during a vaccination response, is to induce a strong immune memory, and then when a manipulating pathogen, such as the ones that I have just spoken about, comes along, if the immune response here that has been generated is of a high quality and is resilient to manipulation, then you will end up with protection. If the pathogen can manipulate the immune response, the immune memory response, and drive it towards its preferred target, then we will end up with disease. And what I'd like to suggest is a paradigm shift, if you like, in looking at adjuvants, not in the terms of the quality of the response that is induced in terms of primary response, but rather assess adjuvants in their ability to induce the so-called resilient immune memory, which can withstand manipulation by pathogens. And that's really what we have tried to be doing in the last couple of years or so. So let's first look at, and I don't need to remind this audience of the classical different T helper types. They will make cell-mediated responses, humoral and immunity mainly against extracellular parasites, for example, and Th17 cells which have their own. And of course, a lot of cross-regulation happens between those different cell types, so that if you push the immune response one way or the other, so the paradigm that you will stay in those pathways. The question is, once you have induced, let's say, a Th1 response, how strong is that Th1 response in the face of a manipulating pathogen that comes along and that wants to push the response to, let's say, another type? And to explore that, we have been using OT2 mice, and for those not familiar with OT2 mice, they are T-cell transgenic mice that respond to one particular peptide of ovalbumin. It's class 2 restricted, and what you end up with is a very high frequency of CD4 T cells, all responding to this one peptide. Those are also CD45-2 positive, so you can track them if you inject them into a different mouse background. So it's a source of antigen-specific naive T cells, if you like, that you can differentiate into different cell types. And that's exactly what we have done in vitro. We can push those ones in different categories using cytokines or ovalbumin as a source of antigen to stimulate them, and on IL-2, we polarize those cells to a Th1 phenotype. If we use IL-2, IL-4, and antibodies to interferon gamma, they go to a Th2 phenotype. If you use another set of cytokines, you can push them in culture towards the Th17 phenotype. And then what we're going to do is to test whether they are really what they are, and in general they are indeed what they expect to produce, with the exception of the Th2 cells, which somehow also produce some interferon gamma, and we don't know why that is. And those are the memory cells that we're going to use to test our hypothesis. And so here you have some of the data. This is the T-cell receptor and CD4, and so you can see that a large proportion are in that category. Those cells make the cytokines that you expect, with the exception of the Th2, which makes some interferon gamma, but otherwise it fits the paradigm quite well. They make IL-4, IL-10, and IL-17, a pretty clean system. Now what we're going to do is to get those cells, inject them in vitro, inject them in vivo, and then stimulate those mice with either quillet, which will stimulate both Th1 and Th2 responses, or alum, which will push the response only to a Th2 type. So this is what we've done. We've taken those cells, differentiated them in vitro, showed that they are different cell types, or used naive OT2 cells as a control, and then injected them. 24 hours later, we will stimulate with either OVA alone, OVA with alum, or OVA with quillet to push the response, and see in how far those cells that we have differentiated in vitro here are able to resist manipulation by those different adjuvants. So those are the results. For interferon gamma, we can see that effectively the cells that produce interferon gamma continue to produce them whenever they are stimulated with either OVA, quillet, or alum, and there's not a very big difference. They all make the same or similar levels of interferon gamma, with the exception of the Th1 cells here, which were stimulated with quillet. The picture is much more clear with IL-4 and IL-10. When we have naive cells, they don't produce any IL-4, IL-10, whatever you stimulate them with. Th1 cells, whatever you re-stimulate them with in vivo, you cannot get them to produce any of those cytokines. Th2 cells, on the other hand, will be producing lots of IL-4 and IL-10, effectively with whatever cytokine you happen with whatever adjuvant you happen to want to hit them with. In other words, the cells that we have put in there, the Th2 cells that we have put in there, are continuing to produce IL-4 and IL-10, irrespective of what stimuli you're going to give them. Similarly, with the IL-17, only the IL-17 cells will produce large amounts of IL-17, and that's irrespective of whether you hit them with OVA alone, OVA in quillet, or OVA with alum. In other words, the cells that have been differentiated in vitro are resilient to being manipulated with the adjuvant in the secondary response in vivo. So those experiments demonstrate that if you push the cells in vitro to fully differentiate and then put them in vivo, they will continue to retain their phenotype. And that's a good news for the vaccine development, because it means that if you are able to induce cells and fully differentiate them, they will remain resilient to future manipulation. Now, what's happening in vivo, that's much less clear. And to do that, we have used a sheep lymphatic cannulation system that they will introduce in the next couple of slides, where we look at the skin and we look at the draining lymph nodes, and preparations injected in the skin will drain to this lymph node, activation of the lymph node will happen, and then we effectively intercept the lymph as it comes out of the lymph node and analyze immune responses at this level of a single lymph node. And when we do that, we take a sheep, as here we cannulate, so we insert a small cannula into the draining lymphatics, and when we inject a preparation, it will go straight into this one lymph node. Because we tie off any other lymphatics, all the cells that are activated in this lymph node will go into the bottle and be prevented from recirculating, which means that both sides of the animal are now independent small immune systems that don't interfere with each other. And effectively, each time we do a cannulation surgery, we intercept the flow of cells from the lymph node and prevent them from going around in the animal and influencing the other side. So two small immune systems can be analyzed in this one animal, which allows us to compare the left and the right side quite effectively. So let's have a look at what happens in a system like that. For example, when we have primed the animals with DNA, we can see a classical second type of immune response. It's rapid. Within 3 or 4 days, we can see it in the lymph node. On the other hand, if we have a non-primed naive animal, we can see it takes a lot longer and the response is much slower. We should note here that in this system, we have sharp peaks in the immune response, and that's because we remove all the cells as they get activated. They are collected in the bottle and then removed from the animal, which means that only the newly produced cells are seen in this system, and you get those sharp peaks in preparation. So now we are looking at local versus recirculating memory cells, and the way that we did that was to first cannulate and then inject those animals with the DNA vaccine, and we can then look at resident memory in this lymph node because the cells have not been allowed to recirculate in the animal. On the other hand, if we first inject with DNA and then cannulate, then the cells have had an opportunity between this DNA vaccination and the cannulation to recirculate in the animal. And if you look at the local draining lymph node, we have this local resident memory and the recirculating memory. When you look at the other side of the animal that has not been injected with the DNA, we will only have the circulating component. And we can compare what's happening in those animals. And you can see here a nice secondary type of response in the resident. Resident or circulating doesn't make a big difference. But if we look at only the recirculating in the first side, so on the other side, we don't see any immune response, which means that the two sides are indeed separated when we do lymphatic cannulation. So in order to exploit this system, what we did is to increase the interval between vaccination and cannulation to see where the cells get activated and when they start recirculating. And in the first instance, we took 3 weeks. In the second instance, we took 5 weeks between the first injection of DNA and the cannulation surgery. So in these animals, the cells will have had 5 weeks to differentiate and start recirculating before we intercept them. And those are the results. 3 weeks, the resident, same as this is our positive control. We see a nice secondary immune response. On the other side of the same animal, where the recirculating cells will be, they are not there. So it takes at least 3 weeks for the cells to start moving out of this one lymph node and move to the other side. At 5 weeks, on the other hand, you can see there's virtually no difference between both sides, and the cells now have had an opportunity to differentiate and start moving around before we intercept the cells from recirculating by the cannulation surgery. So the conclusion of this experiment is that it takes at least 3 weeks, but less than 5 weeks for the cells to differentiate into memory cells and start leaving the lymph node and becoming actively recirculating cells. Now, we use this system to look at adjuvant-mediated manipulation, and we are going to compare the left and the right side of this animal. In the first instance, at day 0, we injected the one side here, the right side, with ova and quillet, and the other side with ova and alum, so 2 different adjuvants. And then, 2 weeks later, we re-stimulate the same lymph nodes here with ova and alum, same as. So we just switch the things around, and ova and quillet. So if you noticed on the right side here, we have injected exactly the same thing as on the left side. The only difference is that the sequence has been changed. We inject first quillet and then alum, first alum and then quillet. And if there is immune memory resilience, then you would expect the left side to respond differently from the right side. If there is no immune memory resilience, after just one vaccination, then there is no resilience in the immune memory as induced as yet. So what are the results? And this is the Th1 and Th2 side. You can see that in both the IgG1, the Th2 in sheep is much higher than the other side. But interestingly enough, when we compare the ratio of Th1 to Th2, or sorry, Th2 to Th1, IgG1 to IgG2 ratio, you can see that the curves almost perfectly overlap. And we have done this now in 3 different animals. They perfectly overlap, which means that one single injection with alum or quillet was not sufficient to induce an immune memory that was resilient enough to withstand manipulation with the other side. So the ratio of Th1 to Th2 is not different in both sides. So in conclusion, I'd like to stress that the quality of the immune memory response does matter. and that persistence of this and the timing of this induction is important. In trying to assess the quality of the immune memory, we came up with this concept of immune memory resilience and the ability to withstand manipulation by pathogens, and started to, and we have just scratched the surface there, to look at ways of measuring this immune memory resilience with TCR transgenic T cells in mice, or look at lymphatic cannulation and different ways of inducing resilient immune responses. Now, where we're going to go from here is to see whether 2 injections, for example, are able to induce a more resilient immune memory compared to 1 injection with one particular adjuvant. And in finishing, I'd like to thank my PhD student, Mohamed Naha and Tellini, Alison Avery is a postdoc, and Fiona Sampson. And the work with the recirculating of the immune memory was done by Rob DeRose, who is now at the Immunology and Microbiology Department of the University of Melbourne. Thank you.
So, Jean-Pierre, if I could start, I mean, in the adjuvant world, there's a belief or a dogma that adjuvant only counts at the first priming immunisation. And if you talk to most vaccine companies, they say that you really don't need an adjuvant once you're primed. Does your model say anything about that, about the potential role, if any, of an adjuvant in the secondary response? Because maybe that's why you didn't see a difference, because it wasn't really needed.
So what we are trying to do here is to see whether is it indeed the first time that the animal sees the antigen in the context of a particular adjuvant. Will that fix the immune response to so that it can no longer be manipulated later on? And vaccinologists would love to think about that, to think that that's the case, because that would make their life very easy. You get whatever adjuvant you want, whatever immune response you want to induce, you use the right adjuvant in the first case, and you go on with your life. I think that our experiments demonstrate that that may not always be the case. Particularly the in vivo experiment that we have shown with the cannulation is that it suggests that 1 injection is not sufficient to set the immune response, and that it's still subject to manipulation once it has seen a single dose of antigen with a particular adjuvant.
But the other part to that is QuIL-A is not really a prototypic TH anything adjuvant, to be honest. Would you have been better using CpG, which really gives a very, very polarised response? And if you used alum and CpG, my guess is you might see a completely different picture, because then you really have opposite poles, whereas QS21 is a bit of a all-around adjuvant. I mean, have you tried CpG?
We haven't tried CpG, but that's definitely something that we'd like to look at. Our results, on the other hand, with alum are very clear. If you, because that's quite an extreme Th2 inducer, and therefore you would expect that even when you come back with QuIL-A afterwards, that you would see a different response than if you come back with other adjuvant, and we don't see that. So I think that, but it's true that using more extreme adjuvants would be a good thing to do.
Okay, so thank you very much. Give you your certificate.
Thank you.